So following the first little video lecture, let's get on with the specific steps of the glyconeogenesis. So as I mentioned before, these are primarily step 1, step 3, and step 10 in the glycolysis, which is now converted into step 1 and 2, and 9 and 11 in glyconeogenesis. So the first step is here, pyruvate to phosphoenolpyruvate. So these are actually two steps using oxaloacetate. And the second part is the fructose 1,6-bisphosphate to fructose 6-phosphate and the glucose 6-phosphate to glucose. So for the first part down here, it's a bit zoomed in. You can see the pyruvate is first turned into oxaloacetate via the pyruvate carboxylase, and that is then turned into phosphoenolpyruvate via the phosphoenolpyruvate carboxykinase. So here we have the use of one ATP and one DTP molecule, which is equivalent to 2 ATP. And so that is where we have a lot of energy input into this. And this is then, you have to remember that this is done twice. So this is actually 4 energy-rich molecules that is used down here. The third, or we could say 5 and 6 ATP, is used in step number 5, where we turn phosphor glycerate into 1,3-bisphosphate glycerate. Now this, maybe you can remember that this step is, well, again, it's a substrate level phosphorylation, but the 1,6-bisphosphate is much more energy-rich molecule than ATP. So this is actually building a lot of energy into a molecule down here. So pyruvate to oxaloacetate, it's a carboxylase reaction that utilizes a biotin so it utilizes biotin as a prothesis group in the enzyme to swing the pyruvate around. Now it also captures, or not captures, well, it captures the CO3 molecule that, as you can see here, formic acid from the solution, and that is then used to create the oxaloacetate. So oxaloacetate is a C4 molecule, so it's kind of like building in an extra carbon in a middle product that will then be released again later on in the next step. So in the carboxykinase molecule, this CO2 group is released, and we end up with a phosphoenolpyruvate. So it's a way to activate the pyruvate molecule so that we can phosphorylate that and get the phosphorylation molecule. Well, this is smart or energy rich. Well, you can always discuss that, but at least that is how evolution has chosen to do this. Now, as you can also see here on this slide, the first step, or both steps, are inhibited by high levels of ADP, which makes sense because if you have a lot of ADP, you don't have that much ATP or GTP, which means that you cannot actually convert this. And this is the regulatory step of the glyconeogenesis. So the first 2 steps here. Again, it's only running if we have high levels of ATP. Now I mentioned that biotin is used as a prothesis group in this carboxylase. Now biotin is a swing arm. So what does a swing arm mean? So we start with biotin down here. This is then bound to the formic acid together with the bicarbonate and ATP, and this part is bound to the amine group here, which is then taken from this situation and swung over to the pyruvate. Now, how does this actually look? So we would have a a pyruvate molecule here, and that would be situated inside the enzyme, something looking like this. And then we would have this arm out here swinging around. So the first step, it would then be in this position where it will capture the CO2. Let's just call it CO2 because it's, or CO3 minus O. So that will be captured up here. Now, what will then happen is that one has to imagine that this part will then move in this direction inside the enzyme, and then it will attach itself to the pyruvate down here. So that's kind of like how one has to imagine the swing arms. One thing that is missing on this drawing is that usually the enzyme in this situation actually goes around here. So there's another domain of the enzyme up here where we will have the binding of the formic acid to the swing arm. So this is the actual reaction, and that's how the pyruvate is then gets the CO2 group on. This is a figure from one of our old textbooks where it's probably a bit easier to see what I just tried to draw before, but this is the swing arm will then move from the BC domain and over to the PC domain in this direction. So the BC is the bicarboxylate domain and the PC is the pyruvate carboxylate domain. So this is where the swing arm will actually work like this. And the middle domain is not we'll try again. The middle domain is not really involved in this part. So how can this actually be going on, and where is it taking place? Now, as seen on this slide, this oxaloacetate formation takes place in the mitochondria. Now, this will make a lot of sense when we've had the lecture on the TCA cycle. So this is in principle a way to sidestep parts of the TCA cycle, but it's also related to the regulation of the TCA cycle. Now, oxaloacetate is a molecule where that can be regulated via the mitochondria. So there's a mechanism where oxaloacetate, while inside here in the mitochondria, can actually not pass over the membrane to get out. So not only do we have oxaloacetate as a middle step, it's during the process the mitochondrial malate dehydrogenase makes it into malate that is then transported over by a proton gradient, and then the malate is then reconverted into oxaloacetate. So this is the oxaloacetate malate regulation mechanism, and of course it only occurs in eukaryotic cells. So bacteria don't have this step to transport the oxaloacetate, but in bacteria it's the same step. So bacteria also have the pyruvate carboxylase and the phosphoenol pyruvate carboxykinase, but they have it all in the cytosol, whereas in eukaryotic cells we have the mitochondria and the pyruvate carboxylases in the mitochondria. It's a way to regulate the mechanism again. Also, it's also a way where the ADP levels plays in, and we'll get back to so ATP and ADP is usually synthesized here on the inner membrane of the mitochondria. So there's a lot of ATP level control in the mitochondria. So it makes a lot of sense that the pyruvate carboxylase, as we just saw, which is regulated by the ADP levels, is also present in the mitochondria. So pyruvate to oxaloacetate, we just discussed that. That is then via NADH transported into malate. That's malate is transported across the membrane again into the cytosol. And here the malate dehydrogenase turns this into phosphoenolpyruvate. And here we can have, one can observe that there could be called upon, at least for the glyconeogenesis, an NADH cycle, so that the NADH that is generated here from the malate dehydrogenase is reused here in the glyceraldehyde 3 phosphate dehydrogenase that we'll come into later. But now we are more focusing on to the phosphoenolpyruvate, and that's where we are right now with these steps. So the PEP carboxykinase, or the phosphoenolpyruvate carboxykinase reaction, looks a bit like this. So what's the first step of the reaction is that we first get a cleavage of the CO2 group from the oxaloacetate. That means that we get a CC bond break here, that then gives us a phosphoenol intermediate, or it's not a phosphate, it's an enolpyruvate intermediate. And then by the GTP coupling, we get the phosphorylation of the middle carbon, so that will be C2 in this situation. That gives us the phosphoenolpyruvate that we know from the glycolysis. So the actual reaction is quite energy saving. So this reaction only requires 0.2 kilojoules per mole, whereas so that's a total carboxylation and phosphorylation of pyruvate, whereas the reverse reaction, so from PEP to pyruvate, actually released the minus 31 kilojoules per mole, as you maybe remember. So here we actually saving a lot of energy by using this carboxylation to make oxaloacetate and then decarboxylate that. So that's a way, that's one of the reasons why it's a bit more complicated than going the other way around. So this number with the pyruvate PEP is of course for the glycolysis reaction, right? So if this is the opposite reaction, so that's where it is. As mentioned before, GTP, ATP is about the same, and in many bacteria they actually use ATP and not GTP. And this should not really confuse you, but most teaching books in biochemistry are written for eukaryotic cells because they are focusing on humans. But most people, a lot of people in engineering of some sort, will also enter into actually engineering E. coli. So therefore, it's a bit interesting just to know that in bacteria, it's ATP and not GTP. And this can also be observed here on the crystal structure of the phosphoenolpyruvate carboxykinase, where in the crystal structure, you have the ATP sitting here, and you have the magnesium and manganese iron sitting here, and the pyruvate is sitting in a bit different position down here. Manganese and manganese can be substituted with each other, but this is the preferred solution. And you can see if you substitute magnesium up here, you actually get a bigger iron, so they will push the ATP in another direction. And possibly it will have a slightly smaller or slightly less efficiency if you have two magnesium ions inside the molecule. So that was the first two steps of the glyconeogenesis. The rest is the same as the glycolysis, so we'll not spend so much time on those. And then now follows the final two specific steps here of the glyconeogenesis. So we get into the glyceraldehyde 3 phosphate. We already used all the ATP we need to use. So at this point, this is 6 ATP, or well, 4 ATP and 2 GTP, but 6 in total. And then we have the fusion, which is the aldolase that fuses two of these molecules, glyceraldehyde 3-phosphate, in, into the fructose 1,6-bisphosphate. So the fusion actually utilizes one glyceraldehyde 3-phosphate and the dehydroxyacetone phosphate, as we've seen before in the glycolysis. So it's basically the totally opposite reaction. So we end up with a fructose 1,6-bisphosphate molecule. Now this has to be dysphosphorylated, and that should be pretty easy, right? It's just a hydroxylation, boom, and it should go off. Now let's see how this works. So, as mentioned before, the enzyme, the fructose 1,6-bisphosphatase, is regulated the same way as the phosphofructokinase. So as you can see here, citrate is an inhibitor of the kinase, and it's an activator of the phosphatase. And this opposite way with fructose 2,6 and the AMP and ADP levels. So in glycolysis, again, this is a regulatory element where we can regulate the production of glucose. And this is also seen, so this fructose, the fructose 1,6-bisphosphatase, this regulation that we observe here is also seen when the source of the carbons for glucose is not pyruvate, but from glycerol or the CO2 that is captured in the photosynthesis. Then both citrate and the fructose 2,6-bisphosphate have an influence whether we go up or we go down on this slide. So this would give us the cleavage of the 1-phosphate would give us the fructose 6-phosphate. The fructose 6-phosphate is summarized into glucose 6-phosphate, as you can see over here by the summarize. So that's on your right-hand side. And then the final step is the phosphorylation of glucose 6-phosphate into glucose, so that we basically get a free glucose molecule. So there's a lot of good or bad things about having a free glucose. So therefore, this is of course regulated quite heavily. Now the first part here is there's a transporter T1 that transport glucose 6-phosphate into the ER lumen. And here on the inner side of the ER lumen, we have the glucose 6-phosphatase. So the glucose 6-phosphatase points inside the ER lumen, and in there it hydroxylizes the glucose and thereby cleaves off the phosphate group. Now this is then transported out with a T3 transporter, which transports glucose from the inside of the ER into the cytosol, and a T2 transporter, which transports the phosphate. Finally, there is the glucose transporters that transport glucose out into the extracellular levels, especially in large species like humans. This is highly regulated because this would also indicate that you would transport glucose into the bloodstream and not just having it inside your cells. So the GLUT2 is very much in co-regulation with the other transporters seen here. Now, what is the benefit? Again, the benefit is that we could actually have glucose 6-phosphate. This could also be summarized into glucose 1-phosphate and maybe built into some of the polysaccharides. But if we don't want that, then we can upregulate the T1 transporter and transport it inside together with an upregulation of glucose 6-phosphate. And then we could have free glucose in here. We could basically store the free glucose in here if we don't have an activity of the T3. And then we would get a glucose buildup in here that would not go through the glycolysis. So it's a way to save some glucose, at least for a while, before it will be transported out of the ER membrane. The other thing is that if we transport the glucose 6-phosphate into the ER lumen, this is not automatically transported out of the cells. And why is this good? Well, it's good because we want to save the glucose inside the cells and not having it going outside the cells. One thing you should note here is that the T3 transporter that I just mentioned that was sitting from the ER to the cytosolic and the GLUT2 actually look very much like each other. So these are more or less the same transporter molecule, which makes sense. It's the same molecule they transport. But this is a depiction of how it's built up so that the transporters are actually linked together in what is known as metabolons. So metabolons are where you have enzymes coming together that do different things, but they sit next to each other so that the substrates here, it's glucose 6-phosphate, doesn't disappear into the big soup of big molecules. So I mentioned previously that the phosphoenolpyruvate carboxykinase normally takes place in the cytosol. There are some situations where this actually happens in the mitochondria. And one example of that is if we have a lot of lactate buildup in the cells, then we first have a dehydrogenase that makes pyruvate and that transport a lot of pyruvate across the membrane. And in order to accelerate the whole process, the phosphoenolpyruvate is then alternatively matured. So instead of it's matured to be active in the cytosol, the whole transcription and translation then maturates it in so that gets a small signal on that it should be sent into the mitochondria where it will be more active. So in this way, you save an NADH molecule that would be used to turn oxaloacetate into malate and then the malate into oxaloacetate again. So saving an NADH is important when you have fermentation because there you don't have a lot of NADH, right? So you're actually lacking these things. So that's one way to try to save some energy in this process. It's not a process that well, it happens when you have a lot of lactate inside the cells and not under normal conditions. The last source is the glycerol, which, well, it's not the last one. There's still the photosynthesis, but that's not part of the curriculum. Glycerol is used a lot in biotech because it's super cheap. It's a surplus basically from bioethanol production, and that is going up and up and up, which means the glycerol production is just getting cheaper and cheaper and cheaper. So how can it be used? Well, first we have the glycerol kinase, which makes glycerol into glycerol phosphate. So here we use one ATP to make the phosphate group here, and the glycerol phosphate dehydrogenase that then turns the glycerol into dehydroxyacetone phosphate. And this then enters into gluconeogenesis or glycolysis depending on the levels of the ATPs. So this is very much depending on the kinase, phosphofructokinase activation or inactivation. And this molecule, I mean, it can be summarized into the glycerol 3-phosphate. We already talked about that several times. So this is a conclusion on the specific parts of the gluconeogenesis. And let's go on with the next video lectures.